الآن
It is my privilege to introduce our next speaker, Dr. Mujahid معصار, the professor of neurosurgery and the head of 21 September University, will be presenting. Please join me in welcoming Mujahid.
السلام عليكم ورحمة الله. أسعد الله نهاركم خير وسعادة إن شاء الله. محاضرتي بعنوان Genetics of Brain Tumors. طبعا أنبه إلى أن المحاضرة طبعا لأن كان الموضوع مشوق. احنا بنحضر المؤتمر مع اللجنة التحضيرية والعنادة والسامي. ولا دكتور سامي؟ طيب فطبعا قلنا لازم ان احنا كنيورو سيرجنز وككلينيكالز لابد ان احنا نحشر انفسنا في هذا الموضوع، مش بس كله على اصحاب القرارات والقرارات. طبعا نحن في امس الحاجه الى هذا الموضوع، نحن بالذات بشكل عام اصحاب الاورام بشكل عام في كل التخصصات، ولكننا نواجه صعوبه كبيره في اورام المخ والاعصاب، خصوصا مع محدودية الإمكانيات. وبالتالي كلفت أحد طلاب جامعة 21 سبتمبر، خريجي جامعة 21 سبتمبر المميزين من الدفعة الأولى طب بشري، بتحضير الموضوع وراجعناه سويا. وهذا من إنتاج جامعة 21 سبتمبر، جمعنا المعلومات حيث أن الموضوع مهم. طبعا الوقت والرئيس شوية كلهم بيتجاوزوا. الرئيس قاعد له؟
لا، الرئيس المؤتمر يلتزم بالوقت.
طيب هذا حس أصحاب عملاء. طيب فشكرا لكل من ساهم في هذا المؤتمر وفي هذه المحاضرة طبعا. وأنبه إلى أنه أول سلايدين هن مهمات جدا بالنسبة للعامة والموجودين كذلك. بقية السلايدات هي شوية هندسة فضائية. ف brain tumors introduction. The brain tumors are abnormal growths of cells within the brain or surrounding tissue, such as the meninges or cranial nerves. They are broadly categorized into either primary or secondary, as any tumor in any location of the body. The primary brain tumors, this originate within the CNS. They are further classified by their histologic types and the WHO classification, which grades tumors from 1 to 4 based on their malignancy and growth rate, concerning the primary. But the secondary, which is the most common, is metastatic tumors. This results from cancer spreading to the brain from other parts of the body, for example, the lung, breast, or skin. These are far more common than primary brain tumors in adults. So we have primary and secondary or metastatic. هذه أهم سلايد طبعا بالنسبة تهمنا احنا كبوبيوليشن، حيث أنه في تجميع الدراسات في الستاديز that between the year of 2015 to 2022, the average annual age adjusted incident rate, which is AAIR, of all malignant and non-malignant brain tumor and other CNS tumors was 26 per 100,000 of population. يعني من كل 100,000 معانا 26 واحد مصاب بأورام الدماغ. The AAIR of malignant brain and other CNS only was 6.8 per population versus 19.19 per 100,000 population for non-malignant. من 26 يوجد 6.8 تقريبا 7 من كل 100,000 مصاب ورم دماغي خبيث. وبقية النسبة يعني لحسن الحظ ولرحمة الله هناك 19 من كل 100,000 من عدد السكان مصاب بالنون مالجنانت اللي هو الأورام غير الخبيثة. In children and adolescents till age of 19 years old, the triple AIR of malignant and non-malignant brain and other CNS was 6 per 100,000 population. The overall incidence طبعا من حظ النسوان ان الاورام عندهن اقل الخبيثه. The overall incidence rate was higher in females compared to male 30 versus 22. This is طبعا في الرجال 30 الى في النساء 30 الى الرجال 22 لكل 100,000. But يعني in the primary malignant tumors more frequent in males. هذا طبعا سوء حظ الرجال. than females 8 إلى 6 لكل 100,000 population. هذه أهم سلايد عامة. Now, epidemiology of CNS tumors. Approximately 26% of all brain and other new CNS tumors were malignant. 26% من إجمالي الأورام إلى malignant اللي هم تقريبا الربع. و74% 74 were non-malignant, making non-malignant tumors more than twice as common as malignant tumors. Gliomas, نوع الأورام الgliomas, accounted for 22% of all tumors. The most commonly occurring malignant brain and other CNS tumors histopathologically was glioblastoma. Glioblastoma is the most dangerous brain tumor, and some literatures tell us that the glioblastoma is the most dangerous or malignant tumor even in the body, the whole body. Glioblastoma. 14% of all tumors glioblastoma. أيضا تمثل 52 of all malignant tumors in the brain or CNS tumors. The most common non-malignant histopathology was meningioma, الأورام السحائية. و contain 43% of all tumors بحسن الحظ طبعا. But including all malignant حتى المننجيوما is malignant. And 57 of all non-malignant tumors. Meningiomas هذا طبعا ورم النساء أكثر شيء. Meningiomas was more common in females than males, خصوصا في البريجنسي. طبعا we are depending on WHO classification of the brain or central nervous system tumors. The 5th edition of WHO classification of tumors of CNS, which called WHO CNS 5 classification, published in 2021, as shown in this very bad crowded table. But tumors are classified by types, some of which are divided into subtypes. Some subtypes are actual variations of the parent tumor, while others are just different grades of tumor, the same name but other or different grading. We have two suffixes are used commonly in this classifications: the NOS or NAC. NOS, not otherwise specific or specified, indicates that molecular testing was either not done or that the result was uncertain. يعني النتيجة ما بتكونش محددة أو حتى معروفة، وبالتالي it is not 100% diagnosis even with molecularity. NEC, none elsewhere classified, is used when there is concordance between the histologic features and molecular testing, or when results show non-canonical result. For example, might occur with a emerging new types. We are discovering every year new type of tumors. So these are the classifications differing between the brain tumors. It's overcrowded table. We have a lot of types. We are the red typing is the most important thing. Subclassifications. The gliomas and the glioneuronal tumors and gliomas is very variant. Choroid plexus tumors. Choroid plexus tumors. طيب choroid plexus tumors and embryonal tumors, pineal tumors, cranial spinal nerve, meningiomas, other we have a lot of variations in tumors of the brain. Just a sample of brain MRI or CT scan with different imaging studies. So, genetics of brain tumors in WHO CNS5 molecular genetics plays the central or the central role in the classification of CNS tumors. Even with our histopathological results, we are not dependent on them. The genetics is very important in the classification. So, the molecular alterations are integral to accurate diagnosis of CNS neoplasms. Sometimes the histopathology result was showing the result as a benign, but it actually is malignant. Table 2K, the molecular alterations that are involved in the diagnostic classification of major CNS like this. This is the table of major classification. The head of the tumors names, adult type diffuse gliomas, pediatric low-grade gliomas, pediatric type, high-grade circumscribed astrocytic gliomas, neuronal and neuronal tumors, ependymal tumors, embryonal tumors, and meningiomas. We have a lot of subtypes in the second column, and we have the molecular drivers of genes. So, how we are categorizing or dividing the brain tumors genetically. Categorizing brain tumor genetics by biological functions is most effective way to understand the complex molecular landscape of neuro-oncology. Rather than just memorizing a list of mutations, probing them reveals how tumor hijack normal cellular processes. So broadly, the genes driving brain tumors fall into five major categories. As we will say, this is the five major categories, ruling of division or grouping or categorizing brain tumors. Number one is the epigenetic modulators oncohistones. Number two, the growth signaling oncogenes. Number three is tumor suppressors or cell cycle regulators for telomere maintenance. 5, DNA repair. In each category we will discuss briefly. We will start the number one, epigenetic modulators oncohistones. There is no oncogene in this type or the class 1 or type 1. No oncogenes. But the modulators, the epigenetic modulators are proteins and molecular systems that govern the epigenetic landscape essentially. It is a software that tells the genes to turn off or turn on the changing the genetic code without changing code itself. It means, right, this don't change the genetic code itself, but dictate how genes are expressed, either stimulators or inhibitors. The most common one, type 1 or enzyme, IDH. IDH1 isocitrate dehydrogenase. Normally, this enzyme helps with cellular metabolism. When mutated, most commonly in the position of R132H, it gains a toxic new function. It produces mutant metabolite called 2HG. This chemical fluids the cell and paralyzes the enzymes responsible for removing methyl tags from DNA. So it results or causing massive hypermethylation that silences normal functional differentiation genes defining features of adult. This type of tumor is adult low-grade glioma. The genetic modulators, as we see in this table, we have a lot of, or for examples of gene category, like number one, metabolic epigenetic, IDH, have a lot of example genes, and the category oncohistones, and chromatin, associated with a specific type of brain tumor, adult diffuse gliomas, diffuse midline glioma, or diffuse hemispheric. So we have a clinical significance of each gene and the categories, like the oncohistone type 3, seen in adolescents, mimics GBM but has distinct epigenetic signature. Type 2, which growth signaling, here we have oncogenes. First one, there is no oncogenes, just signals or just modulation of response. Growth signaling oncogene, it is an oncogene, a mutated gene that drives the uncontrolled growth of division, as we see in the picture. The normal oncogene stimulate the proliferation normally, but the mutated oncogene leads to abnormal cell proliferation, leading to tumor or mass. It converts from, it is normally a proto-oncogene, which are normal genes responsible for essential cellular functions like growth, signaling, and survival. But the proto-oncogene is changed so that too many copies are made, so it is become more active than normal, which become oncogene. The most common and dangerous oncogene example is EGFR, which is very common in our field of neurosurgery. This is a receptor, the receptor of epidermal growth factor receptor. It is a receptor receiving growth factor in outside of the cell that leading to a growth or massive classification or differentiation of cells leading to tumor. We have a lot of genes with specific mechanism associated tumors and clinical impact or surgical impact. The number 3, tumor suppressors and cell type, cell cycle regulation. Tumor suppressor genes, TSG, tumor suppressor genes are genes that protect the cell. We have genes that protect, it suppress the tumor, but if this changed, it leads to tumor, to overgrowth. We have 4 types. The most important types that we see in red lines, number 3 and number 4. Number 3 with CDKN2A، يعني حب الاميونولوجي وانت تكتب نبي رواح دكتور نبيل والدكتوره تشوفوا على حروف وعلى لا. طيب CDKN2A and B in IDH mutant astrocytomas, homozygous deletion of these genes are most commonly in chromosome 9p mandates WHO grade 4 diagnosis. Grade 4 in neurosurgery is the most malignant. It's killing. It's a killing tumor. This is malignant. Number 4 in F1 and F2 طبعا this is commonly used in all subjects of medicine. In F2, myelin loss is the most common driver in convexity melanomas and vestibular schwannoma. Type 4 or category 2-4, telomere maintenance. Telomeres are repetitive DNA sequences بنهايات الكروموسومز at the end of chromosomes that protect against genomic instability and prevent unwanted DNA damage response. In most somatic cells, telomeres progressively shorten with each division, limiting cellular lifespan. However, the cancer cells bypass this limitation by activating telomerase or the alternative lengthening of telomeres, enabling unchecked proliferation and tumor progression. This is a new mechanism. And this type or category very important اللي بيهم الأونكولوجيست زملائنا الأونكولوجيست عليه. Their status often dictates how well our adjuvant therapies, adjuvant therapies يعني after surgery rather than surgery, temozolomide and radiation will work. The MGMT promoter methylation silencing of this promoter is primary predictor of temozolomide. مش معنا احنا في النيوروسيرجري في الجلايوبلاستوما الا كيموثيرابي محدودات جدا، من اشهرها التيموزولامide sensitivity regarding the MGMT promoter. The TERT promoter mutations allow cells to achieve replicative immortality. TERT mutation is a red flag for aggressive glioblastoma behavior. Mismatch repair defeats lead to hypermutant states often seen in recurrent GBM. After temozolomide potentially opens the door of immunotherapy. Still closed, not open yet. Last one or last category, which is the DNA repair genes. DNA repair genes are specialized genes that encode proteins responsible for identifying, removing, repairing damaged DNA, maintaining genomic stability, preventing cancer. These genes manage critical repair pathways, including base excision repair and nucleotide excision repair. Fixing lesions caused by metabolism, radiation, and toxins. Defects in these genes can lead to severe disease, including in hereditary cancer. In humans, في البشر بشكل عام, we have 125 genes that have been identified as being directly involved in this category, this repair process. Most important example, as you see in all types, is glioblastoma. Glioblastoma MGMT promoter methylation. The role, while not a cause, it is not causing the tumor itself, but the most important one regarding GBM has methylated MGMT promoter. Cannot repair the damage caused by chemotherapy drug. زي ما قلنا بيهمنا في treatment with adjuvant temozolomide, significantly increasing the patient's survival time compared to those with unmethylated active repair. The summary of these 5 types is number 1, epigenetic oncogenes, as we discussed before, tumor suppressor, telomere, and DNA repair. With the representative genes commonly طبعا بيهمنا احنا كنيوروسيرجنز كاونكولوجيست كديagnostics in histopathologies. Key tumor example, the astrocytoma, glioblastoma, and oncogenes, meningiomas in tumor suppressors, oligodendroglioma in telomere, and glioblastoma mentioned. Glioblastoma mentioned in every category. DNA repair. طبعا بعد ما خلصنا هذا الشرح الفضائي بالنسبة لنا احنا ما هو المطلوب؟ الآن خلصنا الشرح، ما هو المطلوب بالنسبة لنا في اليمن في دولة محدودة الإمكانيات، وطبعا دائما نحن في خلاف دائم مع أصحاب الهيستوباثولوجيز ومع ما بنختلفش احنا والأونكولوجيست صح؟ فدائما في تعاون، وإنما اختلافاتنا دائما تجي لنا الهيستوباثولوجي ناقصة. بيقول لنا انه خلاص the suspicion of low grade glioma, suspicion of we need grading to start the proper management. So we have recommendations regarding Yemen. طبعا الدكتور شكري يقدم هذا recommendations طبعا، ودكتور شكري هو من خريجي جامعة 21 سبتمبر الدفعة الأولى، وهو الآن في سنة أولى نيوروسيرجري في المخ والأعصاب، بورد يمني وعربي. فأقدمه لكم وقتي في وقتي. خلصت نص الوقت. تفضل دكتور شكري.
صباح الخير وسعيد ان انا متواجد معكم اليوم. I will talk about recommendations according to brain tumor and genetics. So as mentioned by Professor Dr. مجاهد معصار, the WHO fifth classification histopathology doesn't play a major role. Genetics play a major role. But how can we diagnose brain tumors in Yemen with limited resources? According to the WHO, we can approach a tier category or a tier approach to diagnose brain tumor into three systems. The first tier is which is available almost in every laboratory. As example, we can search isocitrate dehydrogenase, our marker. We have isodehydrogenase H1R100H. This is the most critical marker for adult gliomas. A positive result confirms the presence of the ADH1R132H confirm the DH mutant astrocytoma or oligodendroglioma. ATRX, dose of nuclear expression, combined with the ADH mutation, identifies astrocytoma ADH mutant. So, if we reach a diagnosis by immune histochemistry, that is good. If we didn't reach, what's the next step? We can do a PCR testing, a targeted molecular testing. So, if that has real-time PCR, some laboratories and we can patient has ADH mutant, and we couldn't identify it by immune histochemistry. For example, we couldn't identify ADH1R32H. We couldn't identify one by PCR. Negative, and he is less than 55 years old. We do PCR. If it is positive, it is identified ADH mutant. Another example is MGTM promoter method. Predictive marker. If glioblastoma patient will benefit from temozolomide, خلاص انا بدون ما اجرب كيموثيرابي على بيشنت، كيموثيرابي غالي والافيلابيلتي غاليه مش موجوده، ممكن ابدا اجرب بالبي سي ار ال جي تي بروموتر. If it is positive, we can start temozolomide therapy. If the PCR is negative, or we diagnosis by immune history or PCR, we can do transportation transport by formalin mixed paraffin علشان الكل يستفيد. الموضوع مهم العربي علشان الكل يستفيد. طبعا قلنا WHO classification معدل على الهيستوباثولوجي، تعتمد على المولكيولر تيستنج اكثر. فشخص الامراض حق البرين تيومرز باليمن لازم نعمل 3 ستيبس. اول ستيبس اميون هيستوكيمستري او الكيمياء النسيجيه المناعيه، وجبنا اكزامبل عليها اللي قلنا الاي دي اتش ار 100. اذا طلعت بوزيتيف ايدنتيفايز استروسايتوما او الامراض او ايدنتيفايز الجلايوما او الاورام الدمويه. اذا كانت الاي تي ار اكس لوز نيوكليير اكسبريشن او فقدان التعبير النووي مع الطفره الاي دي اتش ايدنتيفايز استروسايتوما اي دي اتش ميوت. قلنا اللي هو الفحص الجزيئي المستهدف البي سي ار. اذا كانت نيجاتيف الاي دي اتش 132 الاي دي اتش 1 اور 2. المستوى الثالث زي ما قلنا اذا ما فيش افيلابيلتي البي سي ار او النيجاتيف دايجنوستس we can do اللي قلنا عليها الحاله الاقليميه ننقل الانسجه اللي تكون فيكس بالفورمالين وامبيدد بالبارافين.
شكرا دكتور مجاهد ودكتور فلاح. تقريبا الوقت اخذ اكثر لانه الساعه قلبت، لا الساعه بدات وبعدين قلبت قلب مره ثانيه. كانت بدات 15 بعدها طلعت 20 مش عارف. المهم يعني الدسكشن لو في شيء نأجل إلى آخر السيشن.